No, it's Mary. Sorry. So I don't know if I should introduce, but might as well. I see some people who have not met her yet. So Janine's an associate professor of radiology at WashU, St. Louis. She obtained her PhD in neuroscience from the University of Sheffield, and then did a postdoc at Oxford with Sonia Bosch and Steve Smith.
Mm-hmm.
So her research program lies at the intersection of AI, mental health, and computational neuroscience. Especially, she's interested in looking at large scale databases, public data sets like the HCP, the UK Biobank, and how to harness their power to analyze complex questions such as heterogeneity and depression, for instance. So before I met her, I actually knew of her through her textbook, fMRI Connectivity, part of the Oxford Neuroimaging Primer series. For those of you who actually were starting out in the field, these are really good, great books. Everything you need to know about that research topic in like maybe 40 pages or so. And I've actually read them and it's been very helpful. She's also an advocate for open science and as a handling editor for the Journal of Imaging Neuroscience, and has served as past chair of the Open Science Special Interest Group at OHBM. So today she'll talk to us about, again, harnessing large data sets for depression heterogeneity modeling and challenges in analyzing these large data sets and her solutions to these problems. So welcome to the
Thank you. All right, well, thanks for the invitation. I was trying to remember on the plane how long it had been since I'd stepped foot outside of LAX to go into LA, and I think it was a while. So it's nice to be back here. And thank you for those who actually made the way here to come in person. Like, please, I'm happy for this to be interactive, so ask me questions anytime. And also, of course, those on Zoom, I get it. I do it all the time. And we'll try. Are you paying attention to the Zoom chat if there's any questions from that? Oh, okay. I thought this was... Right. This is picking me up, right? I don't need to be... I don't like to hide. I like to, like, you know... The mic will, exactly, yeah, okay. So yeah, I'm gonna talk about big data. And so I thought this is kind of a funny figure that I created. So each dot is a paper that I'm an author on. And so it's plotted as a function of years of publication and then sample size as a log scale. So you can see just, I like to make the joke too, when I'm teaching, like I did my first study in 25 people. And now I have like tens of thousands. And I know I'm old, but I'm not that old yet, all right? And so I still think that's pretty cool and pretty amazing. And so I'd like to tell you today about some of the things of how we use those bigger data sets. This is a table from a review paper that we published. And so I'm gonna be on the sort of right end side of this. So I'm interested in, like, ideally epidemiological data sets where we don't have a lot of inclusion-exclusion criteria and we're trying to get a population representative sample, a little bit in the retrospective big data. It's kind of interesting that big data is also happening at this sort of left-hand side of the scale where people are scanning few subjects, but lots and lots of times. I'm not gonna talk about that a lot today, but I think it is quite interesting to see sort of the focus on both ends of the scale. And so happy to discuss that if anyone has experience or is interested. But I'm interested in individual differences, how brains differ from each other and how that maps onto other interesting things. So that's why I think that right end side of the scale is where I want to be. So you probably, I'm actually going to talk about a few different data sets, but a lot of the work that we've been doing is using the UK Biobank. I'm sure a lot of people here have heard of it. They've now finished imaging in 100,000 people. Again, I think that's still so cool. And this is not a healthy data set. You know, the incidence of depression matches the incidence of the overall population. And so it is what's called a prospective epidemiological data set, meaning that people are getting scanned, they're sort of middle to older age, they get followed up until they pass away, and all of the health records are available. And so it should be a very rich data set from that perspective. They measure everything. Like one of the measures is how much cheese did you eat in the last week? So I do mean everything. They never quite measure it the way you want it. And so some of our work goes into like validating and making sure that the way that we're measuring depression kind of maps onto other measures of depression. So this is kind of an outline of the talk for today. And so I want to focus on like how do we use kind of these population or bigger data sets. And I think the subtitle of my talk is what works, what doesn't, and what's next. And so I'm going to start off with two different studies that I think that we've done in the lab that I think use population data well in terms of like leveraging its strengths, and give two different examples. The second part of my subtitle was what doesn't. I think I'm going to rephrase that to what's harder than it looks, because I'm talking about my own work, so I'd just be a bit nicer to my own studies. And so I want to give a couple of examples of subtyping and also disease classification in that. And then the last section of the talk is what's next. And so those are sort of more hot off the press kind of things that we're working on right now. Those are some unpublished things. And so I want to talk about data harmonization. This is a project that Catherine and I and others have been working on a lot over the past few years. A little bit about resilience. And then I think Shantanu, you were like, I want to hear about your resting state work. And so I'll end on that with some recent stuff that we're doing on sort of not necessarily depression specifically, but more functional organization and trying to understand that. So yeah, that's the layout. That's the, I think, 7 papers that I want to go over. Again, feel free to interrupt me anytime. Okay, so starting off with what works, and I kind of, we've been, I think when you first get big data and they measure things like how much cheese do you eat, the temptation is to like just throw it all into like one big computational thing. And we did that, and you get something, and it's significant, and it will replicate because you've got so much stuff. But it's hard. We were talking about this at breakfast, actually. It's hard to say anything meaningful from it. And so I think where we've moved now is like trying to leverage the strength of these big data sets, but without trying to also recognizing that there's a lot of weaknesses of these bigger data sets. And so there's a lot of studies that you could do better in a traditional patient control design than you can do in these data sets. And so don't try and do a poor man's job of a study that can be better done in other ways. And so these two studies, I think, in these population data sets, the majority of people don't have depression. There's a lot of subclinical symptoms. There's a lot of transdiagnostic information. In some cases, we're looking across the lifespan. And so I think that's both a good thing and a bad thing. You can study psychopathology kind of in all of its messiness in the real world. And so I think there's two options. There's the sort of embracing just the variance and the bigness of it all, or you can like parse it down. And so the two studies that I'm going to talk about are kind of giving those examples. And so this first study that I'm going to talk about was inspired by some work. So this is a brain map. This is a meta-analysis of depression. And so they find the different studies that went into this found information everywhere across the brain. But if you look at the p-value on the right-hand side, this is actually a null meta-analysis in that they didn't find anything that was consistent across all of the different studies. And this is one of the tables in the paper. And you can see that the sample sizes are pretty small. I think one of the bigger ones is like about 70 patients. And these are realistic. When you're doing studies, it's already hard work. It's a lot of work. But so our question was, okay, if we bring together big data sets and we do something similar, can we start to find something more consistent? So the overarching study here was just, can we find reproducible correlates of depression using big data? This just got accepted in Nature Mental Health, and it was led by 2 trainees in the lab, Cassie Hamilton and Lexi Luo. And so we took 6 different data sets across the lifespan, so the UK Biobank, ABCD, all of the HCPs, and one kind of smaller one at the bottom there that's a UPenn data set specific more to depression. And then in each data set, we selected a couple of depression phenotypes. So one broadly looking at predisposition or risk of depression, and then one broadly looking at severity. So these were all self-reported measures. And then we had a bunch of imaging-derived phenotypes, or just tabulated measures from structural and functional information, cortical thickness, gray matter volume, functional connectivity. And essentially the methods here are really simple. We just did straightforward lots and lots in every separate data set, lots and lots of univariate linear associations, separately for predisposition, separately for severity, separately for all of the different imaging measures that we had. And then we did a meta-analysis to kind of collate those estimates from the different data sets in one. And then lots of corrections across all of the, I think, more than 100,000 imaging phenotypes that we had. And so these are some of the results. So severity is on the left, susceptibility is on the right. These are volcano plots that you can see on the outside. So it's showing meta-analytical estimate effect size on the horizontal axis and then p-value on the vertical axis. And so that means that anything in that bottom left quadrant, let's see if people can, so anything in these lower left quadrants is significant. And then these yellow figures are showing those dots essentially on the surface of the brain. And so that's a lot of information here. So let's sort of break it down a little bit further. So these were some of the overarching results. So these swarm plots now are showing all of those dots in terms of distribution across the different imaging kind of modalities that we looked at. And so we get the biggest effect sizes for structural information, particularly surface area and gray matter volume. It's pretty complicated for functional connectivity. So we saw nothing significant at all for partial functional connectivity. So that's when you're controlling for all other parcels in the brain. Quite a lot for full functional connectivity. We did do some heterogeneity, like meta-analytical estimates for the full functional connectivity, and there seemed to be quite a lot of divergence across the different data sets. So initially we didn't have those in the paper, but the reviewer wanted us to add them in. Yeah. Yes.
Even if this example, that still wasn't like their question. So when you're looking at various CPA, you've not been given the whole. I just wanted to.
Yeah. So for those at home, the first question was, why didn't I include Catherine's data? And it was because we started this project, like this was a big effort. So we started this like more than 2 years ago. So, or I think even 3 years ago. So, yes. And the second question was, we don't have a lot of severe people, which is totally true. So there is, within the UK Biobank, we did subselect about 11,000 people who had some history of depression. So we tried to get, at least in that data set, get rid of some of the overrepresentation of non-depressed individuals. I mean, if you're fitting a linear regression slope, then if the effect size is the same across everyone, then the slope should be the same across everyone, and it shouldn't matter what your severity is. I will come back to that in some other things. I think it's a trade-off between power or sample size and severity, right? Like I'm not aware of a way where we could have had big sample sizes. And actually, you see in some of the analyses that we've done that the UPenn study here had much more variance than any of the other studies because it was by far the smallest.
Yeah, maybe the thing those that you saw into structure were related to severity, but maybe you would have gotten more what you had. That was it.
Yeah. So the Catherine is saying that you might get bigger effect sizes for sicker people. And I think that's a fair point. So yes, the functional connectivity picture is complicated. Some of the surprising results that we found when delving more into the gray matter in particularly, we see that visual and somatomotor, so this is the same result as those results on the left, but kind of broken down into where we are in the brain. And so you see relatively large effect sizes, particularly in somatomotor. You see that that bar is kind of similar to default mode, which is interesting and hasn't been the focus of the literature on depression as much. And so that was somewhat surprising to us. We also see much less involvement of the subcortical region. So we didn't, even though we tried very, very hard, we couldn't find anything consistent, even at the single data set level in the hippocampus or the amygdala, for example. And that might well be related to what you pointed out in terms of severity, in terms of this being a population data set.
Well, doesn't surprise me visual. I think it's one of those things. There's bias, right? Because we, in our reporting, we can have effects all over the place and then focus on the limit.
Yep.
But in our own data, we've seen plenty of kind of visual. Maybe those are also larger, more less heterogeneous,
yep.
as we know. So the effect of that and the limbic are smaller parts of it. But so on both, I think,
yeah.
First of all, they are both more nutrition than people admit to, and then people who benefit from them. Being a bigger stock and more homogeneous.
Yeah. Yeah, so for those on every single conversation I've had with people, it has gone this way where I say, it's surprising, someone a motor, who knew? And then everyone goes, oh, we knew, we see this all the time. And I'm like, well, where is it? I can't find any papers that are really properly talking about this. So, and you're right, it might well be that there's just less kind of variance across subjects in those regions. And that's why it comes out stronger, statistically speaking. So,
so just so you know, analyzing separately the various data sets. Do you have a sense, if you put them all in one giant data set and actually analyze them as if they were one enormous experiment, do you have a sense of what is it, sort of at what point, sample-wise, results converge? Any of these results would converge?
Yeah, so the question is, if we had put them into one large analysis, at what point does sample size not matter anymore, and do those results converge? We didn't do that because each of the data sets were pre-processed slightly differently, and they all had different measures of depression, which we actually thought was a strength. Like, right, if you find anything that's consistent across these data sets, then it's robust against those like real life variations. I suspect, so we did do a fair amount of digging into, like lifespan effect, because we have younger people and older people here. And so we compared data sets and we looked at to what degree effect sizes were consistent across data sets, like correlated effectively across data sets. The UPenn study at 185 people is definitely the outlier. That one's definitely much more variable in terms of and much more inconsistent with the other data sets. For the other one, so it generally, it all just varies with sample size. So the 1,000-ish data sets were more stable. The 10,000-ish data sets were even more stable. So overall, there was pretty good convergence between things. But I don't know if I'd want to put my name behind an exact point. You do still see an improvement in convergence from 1,000 to 10,000, I would say. Okay, so one thing to point out here is that, these meta-analytical effect sizes are pretty small, even for the ones that are significant. So we're not even getting to the 0.1 effect size, which I think is consistent with what other kind of well-powered studies are finding. It's still surprising when you think about depression being kind of a, something in the brain. And so the second study I wanted to talk about, this is actually treating big data in exactly an opposite way. Instead of just like throwing everything in together, here we're trying to treat the UK Biobank specifically as like a database from which we can pull interesting subjects. And so this was a study led by Kayla Hannon. She used to be a grad student in the lab. She's now a postdoc at McLean in Boston. And she was interested in, okay, maybe those effect sizes are small because because different kind of symptoms or clinical aspects of depression have different neuroimaging correlates, and we're kind of washing that all out when we're averaging that all together. And so what she did is she looked at, most people with depression have a mixture of all symptoms, but she looked for people who were high in one symptom and then low in all of the other symptoms. And if you were going to recruit for this study, you could never do it, right? This would take years to do. But when you've got, 100,000 people sitting there, you can find, decently sized groups of these people who happen to like explicitly dissociate between these symptoms. And she did it for some other kind of aspects of depression as well. So for example, chronicity, like how many episodes have you had over your lifetime versus how severely depressed, how severe are your symptoms. Now, those things are usually very highly correlated. But again, you can dissociate them specifically if you have enough people to search from. So this is using big data in a very different way. You can see our sample sizes are, smaller, much smaller than the previous study that I showed because we're kind of sub-selecting and parsing things out. And then we have a heterogeneous group at the bottom, and this is kind of more your typical, like, mixture of symptoms and mixture of information. And so Kayla did normative modeling. So you see normative deviations on the y-axis here, and these were some of the imaging measures that she had significant results for. So those include, actually we do have the amygdala here, some connectivity at the end there, some fractional anisotropy at the start. And so I'll show you the results in a minute, but essentially we had two hypotheses going into this study where one was that, so I'm going to show you for each imaging measure all of the different groups at the bottom right there. And our hypothesis was that each of the sort of really tightly controlled groups would have higher effect sizes than that heterogeneous mixture group. And that maybe in some cases they would be inconsistent with one another. And so I'll show you the results here. There's a lot to take in. You can stare at this for a long time. We have. But I'll draw your attention to a couple of things. So in each of the columns, the last bar is the heterogeneous bar, is that dark red one. And so you can see that the effect size, the bar size of that heterogeneous group is smaller than all or at least several of the individual homogeneous groups. So that confirms our first hypothesis that when you parse this sort of variance that we're seeing really tightly and you really control that tightly, you see stronger effect sizes. And the other thing that you can notice is that for some of these imaging phenotypes, some of these symptoms are going in opposite direction. So you can imagine if you're pooling that all together and looking at just depression as a whole, you might be losing some of that information like we saw in the previous study. So in terms of what works, those are two kind of different ways that I think neither of those studies could have been done with traditional data sets. So those are two ways in which I think, leveraging these population data works. And it is possible, hopefully I've shown you that, to get consistent correlates of effect sizes even across very different data sets. But effect sizes are relatively small, and maybe part of that is this variance that both the size and the directionality of the effect might differ related to different aspects or symptoms of depression. Okay, so what doesn't work or what's harder than it looks is the next section. So I'm going to start off with a, well, I first want to, I get this is a little bugbear of mine. I think when I tell people that I do computational big data research, they think I'm not a real scientist and I just do exploratory stuff and I don't have any ideas going into studies, which I think is not true. Like, it is true, I don't get to decide how the UK Biobank measures something, and I would have made different decisions if it was up to me in some cases. But we do always start with a question, and you will see in the methods, we started off with a basic linear regression. I'm going to end the day on a deep learning transformer, and so we do everything in between, but the method has to fit the question. And we do usually have a hypothesis of what we expect to see. And because we have so much data and the computational tools, we can look at validation or replication. So that's just a little in defense of my research there. But I want to talk about a couple of studies The first study is kind of following on from the previous result that I showed. When you parse out symptoms, you get stronger and kind of diverging neural correlates. So maybe subtypes are the answer. And there's a lot of research looking at depression subtypes, depression biotypes. And so this is another paper by Kayla Hannon. And so her question was, well, people have already published subtypes, but they're hard to compare because they're done in different data sets. So what if we just take like 6 different methods that people have already published on, replicate them as closely as is feasible within our large UK Biobank cohort, and then actually see, do they give you the same answer or not? And so these are 6. So there's 2 that use clinical inputs, 2 that use structural structural MRI features, and 2 that use resting state features. And all of them, and we tried to follow as closely as possible. I mean, this wasn't a replication study. This was a comparison study. So occasionally we diverged a little bit to make the comparison fair. But we did all of these 6 different approaches in the same, I think, 2,500-ish subjects in the UK Biobank that were depressed, met criteria for depression. And so the first news is pretty good. So this is looking at within each method, how well does it replicate? So this is each of the methods separately, and it's doing bootstrapping. So it's asking when you rerun it on 80% of people, do you get the same answers? Do people get grouped the same way? And these results are pretty encouraging. Like there's some variation across different methods, but all of them are internally pretty robust and replicable. Okay, so then we compared them between different studies. And some of these results have a star next to them. Some of these results are significant because that's what happens when you have thousands of people. But all of these are essentially 0. And so this is showing that even though each of these different methods for looking at depression subtypes is internally stable, and we also looked at some validity. So we looked at were there some interesting differences between subtypes, and there were. But they don't seem to agree at all. And they don't even agree within. So we had expected a somewhat of a block design, because remember, 2 of our studies were clinical inputs, 2 were structural MRI, 2 were functional MRI. So we sort of had expected that at least those were much more consistent, and we didn't even find that. So we're still doing some work on this, but it's in the harder than it looks section for a reason. And then we were, to your point, okay, maybe this population business is not useful. Maybe we need to look at like people with a clinical diagnosis of depression. And so that was this next study. So we looked not just at depression in this case, but at 17 different disorders, because why not? And so we got from the UK Biobank the people in the F and G classes of the ICD-10 who had a diagnosis. And then for each of those cases, we had a matched control who didn't have any F or G diagnoses and who was exactly age and sex matched, and I think head motion as well matched. And so here the idea was, okay, in each case we have a set of patients and a set of controls. Let's just use various different, we did this on structural features, functional features, and also socio-demographic features. And let's just see, can we classify patient versus control? And so I'm going to show you the results in a minute. Chance is always at 0.5 because we always have the same number of patients and controls. So, and then those are the results. And so the gray dots are across all feature types that we use. And then the colored dots and the red diamonds are the best performing feature set that we used. And so you can see they're all kind of above chance. But this was not as good as we had hoped it was going to be. I had all sorts of ideas of like, once we have trained the classifier to predict depression, we can test if it also predicts anxiety. We never got to that point because actually the depression one was, so there were 2 that were significant here, which were interestingly for depression and for demyelinating diseases, which includes MS. And it's interesting when you look at the sample size. So depression is the biggest sample size. So you just get a much tighter variance across your estimate, and that's why it's significant. Whereas demyelinating diseases is actually the smallest sample size, but it's a bigger effect size, and so it's your stats 101. You know, your power to detect something is a combination of the true effect size and the sample size. So again, it's not that it didn't work, but again, this was harder than we had expected it was going to be. And hopefully this will be a useful kind of benchmark for future studies using UK Biobank to do this type of disease prediction. Yeah.
Sorry, functional prediction.
Those were the best performing. I think for depression, both structure and function did somewhat equally well for this study. Yeah. In this case, it was just T1, so it was right. So.
Yeah, I mean, this is always interesting to do a case control classification, but like the differential diagnosis of the hard clinically, so like you try like depression, anxiety versus typical, or things that sometimes cluster together clinically that are harder, or I don't know, like this is all you have here, but things that actually would have a clinical reason to try to figure out. It's easy to tell, but difficult, right? But maybe not depression from like bipolar or something. So I mean, did you try to get that harder, even harder class?
We did repeat this where we removed anyone who had multiple diagnoses. So it was only the one unique diagnosis, and the results were pretty similar. Like I said, I had in this project, I had ambitions to do train on one disorder and then test on another to see, which classifiers are like catching something more like kind of p-factor like, and which are capturing really something disease specific. So that was the idea going into it. We never actually got to that. So I would have loved to make it harder, but yeah, this appeared to be already pretty hard. Yeah.
Technical. How do you treat the structural data, meaning when you're realigning it in some common space, you do that linearly, or do you go above 12 degrees of freedom? Because in a sense, that's a trade-off between precision of matching and losing some of the potentially interesting variants.
Yes. So we use the UK Biobank process data, which includes and this was done when they hadn't done free surfer yet, I believe. So I think this is nonlinear volume-based registration, which is somewhere in the middle. But I think it's a really important point, and I'll end on a slide. We're starting to do some precision functional work where I think that question is really, really relevant. I think at some point we were like, we must have coded this wrong. Like this our code is just not working. And so we did the same thing where we had like an older and a younger group. And so we did age. I think I cut it off from this figure, but it's like way up at close to 0.9. Yeah, we initially hadn't planned to do that, but we were like, something's wrong. And so, yeah, we did do that. It is a good sanity check for sure. Yeah. Okay. So in terms of summary, so for at least things that were harder than they looked. So I think depression subtyping, I still think it's a very important question. I think it is not a question that's answered. And I think we can actually learn some of the machine learning literature, like a lot of prediction models, in order to get published, you have to compare against the best existing model, which is not currently something that we do for subtyping in depression, or I think in other fields. And I think that would be quite helpful to do to actually answer this question, because it looks like even things that are reliable and somewhat sensitive, it's no good to a doctor if you get 10 different answers of how to split people up. And so we're actually now starting a project where we're looking at shared latent projection. So we're purposefully forcing a model to put the same subject in the same space, and then seeing if that's a bit more robust if we sort of optimize it that way. The ICD-10 information in the UK Biobank, I think, isn't as clean as people think it is. People think it's like the gold standard for a clinical diagnosis, but there's all sorts of incentives from insurance and from other, doctors are busy. And so I think that data isn't as clean, and that might be contributing to that result, that second result. But we are doing some research on resilience, and I will talk on that actually in the next section. Because, yeah.
And you said you have all these measurements, how much teeth to eat, but there's not that you didn't include that might be critical, like the genotyping, you know, it might relate to advanced. I mean, you know, so there.
Yeah.
But you think you find it.
Yeah. Yeah. No. So the comment was, you know, there might be a lot of information that's relevant to biotyping or subtyping that we didn't include, and we just resubmitted a grant kind of expanding on that subtyping question. So we spent a lot of time thinking about that, because it's, you know, if you include everything, you don't have anything to validate against, because you're going to get some subtypes and you have to somehow say whether they're useful or not. So that's one reason to hold some things out. There also at some point becomes some circularity if you feed everything you know about depression into the subtype. Well, the best predictor of future depression is past depression. So you will get much better predictions, but is it telling you anything about the biology? So I agree with you fully, and I think it's also quite a hard question to decide what to include and what to exclude. Yeah.
Right. I'm inferring, or correct me wrong, that everything you've shown so far is univariate. So you're assessing different modalities separately.
Modalities separately, but like the classifier was multivariate in that each modality's classifier had multivariate features feeding into it. Yeah. Yes. Yes. Yeah.
Whether that might give you a memory.
Yeah, and that's kind of what so the grant that we have under review now, or resubmission, is kind of a competitive AI grant where we're developing multiple different concurrent machine learning approaches informed by some of the things that we know that are challenging. So not just like out of the box approaches, but dedicated approaches. And one of them is a multimodal kind of fusion thing. And the other is a kind of shared latent projection thing to kind of regularize the projections into and then do the subtyping in latent space. So yes, I agree that I haven't given up on it. This is, I think it's just like, okay, we gotta do a better job. But I think there's definitely some things we can still do. Yeah. Right. And if you and you know, a subtyping algorithm will always give you something. So if you force it to use, like you were saying, the information that's shared, then hopefully it will regularize itself a lot and you'll get a lot more stable. Well, you definitely get more stable results, but hopefully they will also be more interpretable and more clinically useful. Yeah. Okay, so last section, I want to talk about these are all very new things that aren't published yet, that are either under review or in preparation, kind of because I like talking about stuff that I'm currently excited and thinking about. So one of them is, and yeah, I will end with the resting state. So the brain representation stuff is away from depression. The first two studies are still in the context of depression. So that's Catherine. This is a grant that we've been working on for a very long time now. And so the idea of this grant is to create, it, to your point, like all of these population data sets are great and all, but most people don't have depression. And so the idea here is that there were 4 studies that are HCP style studies that were acquired kind of in the space of psychopathology, anxiety and depression, internalizing disorders. And they're big, but they're in this context still relatively smallish. And so the idea behind this collaborative grant was to bring all of those 4 data sets into one sort of harmonized data set. This is work that is led by Joe Yarokosemkut. He's a postdoc with me. And so we performed, and Petra as well is heavily involved in this grant. I know many of you know Petra. We performed the exact same HCP pipelines on all of these subjects, extracted imaging-derived phenotypes for all of the different modalities, and then harmonize those IDPs. There's a lot of information I can tell you about, but I'll show you a couple of things. So one of the questions is, how do you harmonize? And this could have gone into like, it's harder than it looks. This whole processing, this whole study was harder than it looks, at least to me, maybe naively. But one of the things we did was compare COVBET and COMBAT-GAM. I know probably many of you are familiar with those. They're relatively simple covariate correction tools that have come from the genetics field. And so COVBET-GAM is supposed to be the newer one, but we did, and we compared that against COMBAT-GAM, and particularly COMBAT-GAM that was trained only on the healthy controls. So we have, from each of the 4 sites, we have about 50-ish healthy controls and about 150, 250-ish patients. And the thing is, the patients differ a lot across the site. So Catherine's patients are on the more severe end, whereas some of the other patients are on the less severe end. So the worry with harmonizing is that you're throwing away the variants that you actually care about when you control for side effects, because they're systematic variants across sites. And so that's kind of exactly what you can see here. So COMBAT-GAM has a functionality currently where you can train on controls and then apply to patients. And so you can see that you remove the side effects in controls, but not in patients, which is gray up there if you're trying to predict site. But that's probably a good thing, because that's information that you kind of want to keep. COVBET-GAM doesn't have that functionality yet. I think Andrew Chen is working on it. But you can feed in psychopathology severity measures to try and keep that variance. But as we've shown here, it doesn't seem to work as well in terms of retaining that variance of interest. There's a lot to be done with this data set. These are just some multivariate prediction regression analyses for anhedonia on the left and depression on the right. And so it's for each of the data sets separately, just showing 3 here, and then for all of the data sets combined at the end there. And so you can, one of the things you can see is that it doesn't necessarily magically get much bigger effect sizes, but you do, we're showing both train and test here so that you can see, I mean, the training effect sizes are inflated because you're overfitting to the training data, but you can see that gap between train and test is smallest in the bigger data set. So you're getting a bit more generalizable, more stable effects, which is what we would expect from a bigger data set. Okay, so I've talked a little bit about resilience. This is a paper that we're almost submitting from Haley Modi, a grad student in the lab. This is an ABCD study. And so the idea was to use social, this actually doesn't have any imaging in it right now. But so the idea was to use social determinants of health. So things like how many friends does a kid have, and what is their childhood experience like, and what are their parents like. Feed them into a machine learning predictor to predict CBCL, so essentially behavioral challenges, psychopathology broadly. And then we calculated, kind of similar to brain age gap, we calculated the kind of residual from the model. So what was the difference between the true CBCL and then the predicted CBCL, so that you can see some people might be resilient, might have lower true CBCL than the model might have predicted, or the other way around. And then we validated that using longitudinal outcomes. So I think we used the K-set so that it wasn't too collinear with the CBCL, but then, and a year later, after all of the information that went into the model, to try and see, like, do those measures of resilience or susceptibility, resilience or vulnerability predict sort of longitudinal outcomes. And these are some of the results. This ended up being a more method-y study. I don't know, I can't help myself, I guess, than we had intended, because it turns out that well, this is again Stats 101, but the model residuals are very highly correlated with what you were trying to predict. So you're always going to have, the same goes in brain age. If your brain age gap is correlated with age. And so if you don't control for that, then you're just going to find whatever, you did a lot of fancy work, and you're just going to find whatever you would have found with CBCL itself. And so you have to control for that. There's various ways of controlling for that. And so we tested all of them. As you can see, without controlling, you get one direction of effects, and with controlling, you get the other direction of effects pretty consistently across all of the different kind of sanity checks that we threw at this. And what our results are showing so far is that resilience actually doesn't necessarily lead to better outcomes. So this is showing that people who, kids who showed resilience at the baseline and 2-year follow-up time point actually had a higher rates of drops in grade. And the same goes to smaller effect size for some other behavioral outcomes. To what degree this is collinearity that you can't really control for, or to what degree this is, you know, resilience coming back to you, you know, you can only show resilience for so long. I think there's still something interesting to be investigated there.
You're not.
Well, the resilience is so the measures that went into the prediction of CBCL were like SDOH, social determinants of health. And then the resilience measure is the residual of the model, so the difference between true and predicted CBCL. Well, I mean,
that, and so now you're putting a label on some of the other measures that,
well,
you know, line up with what I think. Well, it depends on which. We don't have even things. We have like some things that we.
Yeah.
So I guess like putting the label on it is something measured resilience.
Right. Well, I mean, the idea, so there are more and more papers coming out in the psychopathology literature that are doing this kind of gap score analysis and calling it resilience. But yeah, and actually not controlling for this. So they're finding the opposite thing that we're finding, but it's just because in our case, so the model prediction is pretty good. It's, I think, like 0.4, 0.5 correlation between true and predicted, which is, you know, obviously not perfect, but in a held out way, in a valid cross-validated way. So that's pretty good. But the gap score and the CBCL have like a 0.9 correlation. So if you don't control for this, you're going to find exactly the same as if you would have just correlated with CBCL. So I think this, and perhaps, yeah, maybe we should think about a different title, but I think the caution is like, if you're going to do gap score analysis to study this, you got to really think about that collinearity.
In the logical that's what you call it.
Yes.
It's not really true grids and
right. Yeah, it's
you know that don't know. That's what you expect. So you could really be thinking that up, or at least. No, not really.
But they are, the way it's model estimated is that they are better than you would predict cross-sectionally. So you fit a model based on these SDOH values that predicts pretty well across everyone. And then you find the people who actually have lower endorsement of symptoms than the model would have predicted. So cross-sectionally, it is resilience I mean, it's still an algorithmic way of doing it, but it is resilience in the kind of traditional sense. Okay, so a little bit, we have a little bit of time left. So a little bit back to resting state. So when I was working in Oxford with Steve Smith, we did a lot of work on resting state organization, and I'm still very interested in that and active in that. Essentially, resting state data is just like a signal processor's dream. Like it's just a big 4-dimensional, you know, space-time data set that you can, or nightmare. And so you can come up with any sort of algorithm that you want to look at the data and look at networks that people have done. And so this is not a exhaustive view at all. And, you know, some of these methods end up giving you maps. Some of them end up giving you connectivity matrices. Some of them end up giving you movies. And so I think it's interesting. I mean, I'm a geek at heart, so I like the data science opportunity here, but it does make it challenging to like, sometimes I'll look at a paper like that does depression, even in the same data set, the UK Biobank data set that I look at, and I see some resting state result, and I just cannot tell whether they're consistent or inconsistent with my own results because they did something different. And it's, you know, my brain can't translate between those things. And so this is work by Samuel, who is a data science grad student in the lab. This is under review at the moment, where he trained a transformer algorithm essentially to go from ICA spatial maps to Schaefer functional connectivity matrices. So there's nothing to do with depression. There's no behavior in this. It's just trying to take kind of a summary measure and translate it into another summary measure that's derived from the same data, without going back through and kind of redoing the analysis, I guess. So it's kind of trying to solve that problem that I have where I look at a paper and I'm like, I don't know, can you tell me what this would have looked like had they done this, this, and this? It's sort of the idea behind it. So the first thing we did was just to go from ICA maps back to ICA maps, just to kind of validate that the sort of transformer machinery that we're building here is doing what we want it to do, is retaining information. And so that gives us pretty high reconstruction accuracy. So across networks, we're getting about a 0.7 reconstruction accuracy in a sort of held out sense. We're doing reasonably good at translation. So we get around about a 0.4 correlation between true and predicted. It's interestingly much worse for partial correlation. So it's much better for full correlation, worse for partial correlation. But it does translate. So we also did it for the Glasser HCP parcellation. So it does translate pretty well across different parcellations. And interestingly, when you do then, so you can take the sort of translated connectomes that it's giving you and then relate that to behavior to make sure that those predictions are kind of capturing the behavioral variance that you were interested in the first place. And so that's doing pretty well. So we can do that same analysis in a multivariate sense in the measured way, in the light bars, and in the translated information, and you get almost the same results. So that's not, it should work because it's coming from the same underlying data. But that's something that we're excited about in terms of trying to maybe bring together the field a little bit in terms of findings and interpretation. Yeah. We did not. I mean, this is actually an ABCD data. This is the old 5.9 or something release of ABCD data. So it's and we have looked at, I did at some point tell Samuel to be like, okay, we should remove like low quality people to see if our estimates increase, because I was thinking we were just hitting that ceiling of like 0.4-ish, and I had thought that we should be able to do even better. It didn't seem to help much when we removed them, so I don't think that's driving it, but yeah. We did. It's too small. So with HCP we do worse. We have a lower estimate. So we started there and we moved to the bigger data sets. These transformers are power hungry. Okay, and then these are some movies from the work that I did in Oxford, looking at individual variation in network topography as a function of behavior. So on the left-hand side are two different networks and kind of moving through a continuum of behavior, seeing how those networks kind of change in shape and size. We were kind of surprised by that. I mean, we knew there was variation across networks, but we hadn't necessarily thought, like, why does it matter in terms of behavior, right? Where exactly a boundary sits. The movie on the right-hand side is the same kind of continuum, but now looking at the sum of all networks. And so that might explain it. There might be sort of more integration or more overlap across networks at people with kind of more positive. This was that sort of behavioral continuum across lots of different behavioral variables. And so this was old work that I did a few years ago, we are, so I don't have any results yet, but I made this movie and I worked really hard at making this movie. So I want to show you this movie because I'm really excited about this. So this is an active project that we're doing this summer where we're taking, again, in ABCD data, different, because again, the same thing is happening now where people are developing precision methods to look at topography, but all of the methods are kind of, you know, working a little bit differently. And so the idea behind this project is, you know, do they agree? If you have very different optimizations and very different kind of approaches to this, but the same input data, do you get agreement across different methods? And maybe some methods are better than others in terms of test retest and behavior. So that's been so far we've just like processed all of the data to get these maps. So and you know, you can start to see some, like if you stare at these, which I did do on the plane for a while, you can, you know, you saw it there, it breaks up and it breaks up here as well. So you can see, if you squint, you can see that there might be some agreement in some cases. Yeah, that's... Yeah, I mean, the ranges will be different because of the different methods and the different approaches. We are going to compare, this is just a qualitative movie. We are going to spend the summer quantitatively comparing these things, which I think will be interesting to see when they agree and when they don't agree. Yeah, you can. And we'll do like spatial correlations, we'll do dice coefficients, those sorts of things to actually.
But no.
Yeah. Yeah, yeah. And we're actually, this is not all 4 of the, we are actually doing 7 or 8 methods. So this is only some of the methods that I'm showing you here. Like I said, it's an ongoing project, but you know, it's something that I'm interested in terms of like resting state organization. And again, it's this kind of idea of like, can we merge information across different ways that people are doing things, which I think is useful. Yes, I think Samuel's involved in this, and he grabbed some code from the toolbox to develop like the actual spatial, the comparisons. Yeah. But this is essentially a similar idea, but about subject variability. So that toolbox is doing the same thing of mapping things onto each other using group data or group average estimates. Yeah. So it's a lot. We spend a lot of lab meeting time on that toolbox and that paper for sure. It's very cool. Yeah, we like it. Okay, and so I think it was my last slide. So there's lots of people that contribute to this. I'm lucky to have an amazing lab of very smart people and still have some funding to spend on them. So thank you for your time.
Great talk. Questions, but we've already asked this question. I should have asked questions during the talk. Maybe I'll start with one. So the combat GAM and the combat GAM.
Yes.
Generative additive models, GAM.
Yes.
You said one of them is trained on controls and the other ones.
Yes.
And combat GAM is not.
Yes.
Yeah. And that's why there's huge difference between these things. Is that a good idea? I was wondering if that's a good idea just to train on controls and test on patients will always get an extremely high.
Yeah. Yeah. So the question for those online is, you know, the combat GAM versus GovBat GAM question, and you know, one is trained on just controls and the other is trained on everyone. Which of them is a better idea? I think it depends on the data. You know, in our case, we know that the patients differ a lot between sites, and so And really what you're trying to do if you do any flavor of combat is remove the uninteresting variation between the sites, right? Remove the scanner effects but not remove the interesting variation across sites. And so if you train it on controls, those controls are from those sites, and those controls will contain those scanner differences and other site differences that you're interested in. So to me, training on controls is a good idea. And in our case, you do actually, so we did the predictions that I showed you on both versions, and you do see slightly higher predictions for psychopathology when you do the combat only trained on controls than when you do the COVBAT. So I think it's a good idea and a desirable idea. And they are, COVBAT GAM training on controls is a little harder to implement, but will at some point be available. And once it is available, I do expect that will do even better, because that captures kind of covariate interactions multivariate interactions across features as well. So, but that isn't working just yet. Thanks, listener. Bye.
The last few slides, you showed the transformer and SIT.
Yes.
Just wondering, the reconstruction accuracy was 0.73. If you, I know people love transformers these days than GANs, but GANs might give you a higher reconstruction accuracy. But data, I'm not as data hungry as transformers. Wondering what you.
Yeah, I can't. We definitely talked about GANs. I was gonna say we tried that and it didn't work, but I'm actually not. I think. No, we tried Vase, because Vase were also, if you're sampling within, variational autoencoder, like if you're sampling within the kind of latent space, then you might do less well, but you might generalize better and you might improve performance. I don't think we tried GANs, but we have been discussing. I mean, this is a project that's still ongoing for sure. So yeah. Did I? Yeah. Yes. The question is, for the subtyping, did we include both bipolar and unipolar? I believe yes. Yeah. I don't think we explicitly excluded. Yes. We looked at medication, and there was quite small number, like there was only about 1 or 2% of people that were on medication. And so it wasn't enough to like delve into in a lot of detail. I think you got similar results when you excluded those people altogether. So that's not driving things. But yeah, and I don't know. I mean, that seems like a very small percentage of people. So maybe people are just like not reporting medication because it's older people and they're on 20 different pills and they forget. I don't know. So no, this is the UK Biobank for the subtyping stuff. Yeah, no, I don't think it drove. I mean, the bipolar question is interesting. We didn't split it out because there's not the percentages are pretty small. But so I doubt that it would drive anything similar to medication, just because it's when you do a population study, you tend to get healthier and wealthier people. Those are people who have time and access to participate in the study, and so that's definitely the case for the UK Biobank. We definitely need to do that sort of work in more clinical cohorts. And we were talking about big enough ones that are started. Because the thing with subtyping is that even if you start with a few hundred people, you then split them into subtypes. So you end up with like very small groups in subtypes, and then you want to validate them by looking at significant differences between them. And you just don't have the power to do that at that level, even if you start big-ish.
But I mean, the diagnostic information about bipolar, unipolar, and the state that the person is in at the time, I mean, that.
It is, yeah. And we did actually do a similar kind of parsing of things. One paper that I didn't publish was that we did a state versus trait. So we specifically looked for people using the longitudinal neuroimaging data, we looked for people who were like high symptoms in one scan and low in another scan, versus people who were low in both scans but high kind of neuroticism, so high predisposition or risk. That I don't think we found anything for the state. So we were hoping to do like a state, like within subject comparison between a high symptom state and a low symptom state, but nothing survived there.
But I'm in diagnosis, you can measure symptoms on the same day that they were scanned. So you did study and it was probably, I don't know.
They did not very well, but they did measure it on the day that they were scanned. Yeah. Yes. No, but I totally agree. That's what I was saying. Like, there's a lot of things, a lot of research questions that are better answered in other data set. And I think you're stabbing yourself in the foot when you're trying to answer those questions with this type of data set. So we try not to.
One last question about the variance, going back to that variance.
Yeah.
I noticed that the drop in the grade is probably correlated with resilience, and both with and without CBCL measures showed equal effects.
It depends. So yeah, it's the first time I'm presenting this, so I probably went over this a little bit too quickly. So in this case, at the top here, they show opposite effects. But there were some, so the corrected, then you're essentially already correcting for CBCL with that corrected gap score. And then this resilient subgroup, that's a third of subjects that actually didn't have high correlation. So there you also don't get a difference between whether you control for it or not. And those were actually, to me, because initially when I saw this, I was like, well, I don't believe this. It's just a collinearity effect. But because that resilient subgroup actually shows the same effect of like worse grades for a resilient group people, there we didn't really do any statistical correction. That was a group that happened to not have that sort of high degree of collinearity. Yeah, exactly. So, and that's why there's no difference between with or without CBCL, because there isn't a lot of collinearity. So throwing it in as a covariate doesn't explain a lot of variance.
Any other? Let's thank Janine again.
Thanks for coming.